Hi, I'm Evelyn, and I did my project on the topic of microbial antagonism and the effect of human saliva on bacterial growth. This experiment will aim to answer the following question. How do the pre-established cultures of benign bacteria in our mouths deter the growth of foreign strains of harmful bacteria via a process called microbial antagonism? Microbial antagonism is the natural inhibition of a foreign harmful microbe by a more native beneficial microbe. It includes competition between natural probiotics and pathogens for colonization space and nutrients. Certain bacteria also prevent the growth of pathogens by secreting antimicrobial compounds such as lysozymes and glycoproteins. This experiment was based off of Fleming's experiment where he introduced his mucus to a colony of bacteria, and this led him to discover the world's first antibiotic. Pasteur and Joubert introduced a series of experiments where they observed bacteria versus viruses and other bacteria. Based on this information, I predicted that a no-growth zone of 2 centimeters would develop after I introduced 0.5 milliliters of my saliva. I arrived at my hypothesis because of Fleming's findings about mucus and bacteria, and saliva is mostly made up of mucus. The main materials I used were petri dishes, cotton swabs, human saliva, and an incubator. First, I gathered all of my materials, and then I inoculated 6 out of the 9 dishes with bacteria by swiping the sink drain and then rubbing the cotton swab on the dish's surface. Next, using the pipette, I placed 0.5 milliliters of spit onto the center of 3 of the dishes. Then I placed the dishes into the incubator. I observed the dishes at the same time every day for 3 days to look for observations such as the number of colonies growing. Considering that the objective of my experiment was to observe the effect of saliva on bacteria growth, I knew that my independent variable would have to be the introduction of my saliva to a colony of bacteria, so my dependent variable would have to be the resulting no-growth ring around the saliva. I also created many controlled variables such as the same amount of saliva, origin of saliva, location of growth, temperature during growth, amount of time for growth, and type of agar base. During trial 1, I found that the bacteria grew by precipitating from where the dish was swabbed by the cotton swab, and the bacteria from the sink grew along with the bacteria from my mouth. However, there was less bacterial growth in the dishes with the spit than without the spit, which shows that even though the benign bacteria wasn't killing the harmful bacteria, it still induced competition for space and nutrients. This is shown by the graph that illustrates that the dishes without spit had the highest number of colonies. During trial 2, there seemed to be a different type of bacteria growing in the dishes because there were little white fuzzy circles instead of the gel-like substance in the first trial. Nevertheless, the benign bacteria was able to produce a no-growth ring in this particular colony of bacteria, which is shown by the areas around the saliva where no bacteria is growing. The graphs also portrayed that the dishes without spit had the most colonies and no no-growth zones. From these results, one can conclude that microbial antagonism is a natural force induced by competition between organisms and their byproducts. It can also be generalized as it is a selective process since it is not always successful, proving the need for specific immune response. The advantages of my experiment setup were that the incubator supplied the right temperature for growth, the time given for growth enabled a decent amount of growth to see results, and the preparation procedures for the dishes also successfully induced bacteria growth. However, I did make the mistake of swabbing in 2 different ways. The temperature of the experiment was not always consistent, and the measuring techniques I used were very hard to get good, reliable data. This experiment design highly resembled Fleming's early experiments with microbial antagonism. It also relates to similar studies done by Reed McGregor to prove antibiotics' ineffectiveness. Questions that my experiment has left unanswered include what specific antimicrobial compounds were at play during the experiment, and what strains of bacteria are resistant to microbial antagonism. These questions could also be important to the companies who manufacture dental hygiene products, because if our saliva is so important to the immune system, do we really want to sterilize our mouths? My experiment's results could also be used by microbiologists and disease control specialists. In conclusion, microbial antagonism is an active defense agent against pathogenic bacteria through the use of compounds such as lysozymes. However, as shown by the variance in my 2 trials data, it is not effective against all types of bacteria. This experiment proved the competitive nature of the oral cavity and that the mouth does in fact contain beneficial probiotic bacteria. Thus, my hypothesis can neither be refuted nor supported, despite the fact that the experiment definitely established microbial antagonism as a very significant immune defense.